Okay, hello. I hope everyone's doing well. Today's video is going to be about DNA replication. So that is going to be the title of our notes. Oops. And so DNA replication is a process that happens in the cell, and it is just the process where all the DNA in your cell gets copied, and you start off with one um, molecule of DNA, and then you end up with two, and they're identical. Okay, so that's what DNA replication is. That's what it means, is the process of copying all the DNA. I'll say the process of making a copy of all the DNA inside a cell. Okay, so before I get into the nitty gritty and tell you all the details about this process, I just want to first talk about why this happens and when it happens. And so DNA replication, so let me just draw a little picture of a cell. Okay, so here's a cell. And for eukaryotes, there's the DNA. I'm sorry, the nucleus. Here's the nucleus. And so the DNA lives inside the nucleus. And so this DNA replication process is happening inside the nucleus because that's where the DNA is. And so I want to point out that in the previous video, we talked about DNA being a twisted ladder like this. And so it has rungs on the ladder. And so let's just say, for example, if we straightened it out and I were to write some nitrogenous bases on here just to give you an idea of what we're talking about. Okay, so let's just say I'm straightening out the DNA, and let's say that I have an A here and that binds to a T here. Okay, and let's say I have a C here, it binds to a G. Let's say there's another C, and here's a G. Let's say this is a T, this is an A, another T, A, T. Let's say this is just an example of a DNA strand. Let me just put some more bases on here. So this would be G, this would be G, C, T, okay? And so each rung on this ladder represents two bases. So it's called a base pair because there's two together. And so if we were to replicate this DNA exactly, then what we would end up with is another exact copy. Okay, so let me just scoot this over. Okay, and so what we would end up with is an A here and a T here, C, G, C, G, T, A, T, A, T, C, G, G, C, and A, T. Okay, and so our DNA lives in this double-stranded configuration because it protects it from degrading. Okay, so if it was opened up, then the DNA molecule would easily fall apart. But because they are together like that, it keeps it safe and it protects it from like falling apart. Okay, so the DNA replication process happens whenever a cell needs to replicate. Okay, so I'm going to write this down. I'm going to say this process occurs when the cell replicates. Okay, and we're going to talk about cell replication in the coming videos, but right now I just want to focus on the DNA. And so the cell sometimes needs to replicate. So we have this cell, and inside the cell we have a set of DNA with our code, and then that DNA is going to replicate, and this happens only when the cell needs to replicate. And so that entire cell is going to replicate. And so the cell will replicate under certain conditions. And those conditions include things like injury. Okay. So like, for example, let's say I have a pencil, like this pencil, and I stab my arm and I cut a hole in my skin. And so that skin needs to fill in, right? And so whenever you have an injury, your cells are going to replicate to make new cells to fill in whatever hole is there. Okay. So that's one reason for cells to replicate. Another reason is sometimes cells just die. Just like any living organism, there is a lifespan for any cell, and sometimes it just gets old and it dies, and it replicates. Okay, so all the cells in your body replicate at some point. They all replicate at different times, but we'll talk about that in another video. But sometimes cells get old and die. And so that's another reason why a cell would be replaced. And the last reason is when an individual is growing. Okay, so for growth. So if you think about it, you started out as one cell in your mom's uterus, and you became 2 cells, and then those 2 cells replicated to become 4 cells. And then, then you became 1,000 cells, then you became 1,000,000 cells, and then you were like this little tiny fetus that was like the size of a peanut. And then you were born, you're this little baby, like this big. And now you're this huge grown adult. And so, we're made of matter, and that matter had to come from somewhere. And so where that comes from is from cells replicating. So as you're growing, the cells are replicating a lot. Okay? So those are the 3 reasons for cell replication. So let me just put reasons here. Reasons for cell replication. Okay, so let's get into the details of this process. Okay, so what I'm going to do is I am going to list out the whole process. We're going to write each step, and then I'm going to also draw it. So in your notebook, make sure you have room for writing and for drawing. Okay, so I am going to have a subtitle here that is called the process of DNA replication. Okay, and I'm going to explain this in steps. Okay, and so what I'd like to do is each step, how I'm going to explain each step is this whole process is run by little machinery, okay? And they're like little workers, and these workers are enzymes, okay? Enzymes are a protein. These little molecular machines, which are protein, which are enzymes, do all this work. And they're the ones who are doing each of these steps. So I'm going to focus on one enzyme per step, and I will use different colors so that we can, keep them separate. But the first step is that when you have a DNA molecule, okay, so I'm going to draw the DNA molecule over here on the side. It's a double helix and it's all wound up really tightly. So there is an enzyme whose job is to untwist the DNA. And that enzyme is called topoisomerase. Sorry. Topoisomerase is an enzyme who's going to untwist the DNA. So enzyme, whoops, my pen's not working very good today. Enzyme that untwists the double helix. Okay, and so what that looks like is this. So let's say we have this twisty ladder, but now we're going to have a straight ladder. Okay, that's a little bit too wide. Let's just narrow that up a little bit. Okay. And so the topoisomerase is going to take that twisted up ladder and it's going to straighten it out. Okay, because it needs to get access to the nucleotides. Excuse me. Okay, the next step, step number 2, is going to be another enzyme that comes in, and it's called helicase. And the job for helicase is to separate the 2 strands apart. Helicase separates the 2 strands. Okay, and so I'm going to draw that. And so what that's going to look like, I'm going to scoot up a little so we can see, is the 2 strands are held together. Each ladder rung is 2 bases, and the 2 bases are going to come apart. Okay, so helicase is going to open up the strand. It's kind of like unzipping a zipper. Okay, and so on this side, there are certain bases. Okay, and I'm going to draw the matching bases over here. Okay, so let's say this is, I'm just going to draw these so we can kind of understand what's happening. Let's say this is an A, that means this is a T. Let's say this is a T, that means this is an A. Let's say this is a C, that would make this a G. Let's say this is another C, that would make this a G. And let's make this a G, that would make this a C. Okay, because A and T always go together, C and G always go together. And so another thing that I want to point out is that, remember from the last video, that each strand has the opposite orientation. Okay, so this strand over here is going to, I'm just arbitrarily picking a side. It could be either one, because remember, DNA just floats around inside of a cell, so there's no like left or right. Okay, it could be upside down, but we're just drawing it. So this side I'm going to assign 3', and that would mean that this side is going to be 5'. And the other side is going to be in the opposite orientation. So that would make this side 3' and this side 5', okay? Because they're running opposite directions. Okay, so helicase is the enzyme that's unzipping the DNA and splitting up the base pairs. Okay, those 2 base pairs are not held together with a strong bond. They're only held together by hydrogen bonds, which is easy to break. And now, since we're replicating, they need to be separated so we can code each side and make a replica. Okay, so I want to point out that this area here is called the replication fork. Okay, so this area here where replication is taking place is called the replication fork. Okay, so the next step is, now we're going to make some new DNA. And there's a very special and important enzyme that does this. This enzyme is called DNA polymerase. Okay, DNA polymerase is an enzyme that makes new DNA. Okay, so this enzyme makes new DNA. And a couple of things. One is that this enzyme can only build new DNA from a 5' to 3' direction. So I'm going to put only 5' to 3'. Okay, the enzyme can only work that way. And it's based on the fact that this enzyme has a specific shape and it can only build in that direction. It cannot put a nucleotide on the 3' end. I'm sorry. So let's see. It only puts it on the bottom. Okay, so it only puts it on the 3' end. So it cannot put a new nucleotide on the 5' end, only on the 3' end. Okay, that's what I meant to say. So the DNA polymerase can only put it from 5' to 3', the new DNA. And what it's going to do is, this DNA polymerase, there are lots of them. So there's going to be 2 here. One is going to build a new DNA strand based on this left side, and another DNA polymerase is going to make a new DNA based on the right side. Okay, so I'm going to draw a DNA strand. I'm going to use a different color than red. I'm going to use purple. And so the new DNA that's going to be being built on this side is going in this direction. Okay, so this DNA polymerase, so I'm just going to kind of draw like a enzyme here. This is DNA polymerase, okay? You with me still? And so this DNA polymerase is going to add DNA. Okay, I'm going to make the new DNA strand blue. And so here's the new DNA strand that this DNA polymerase is making, and it's making it from 5 prime. So this is the 5 prime that meant to be, that was supposed to be a 5. Hold on. This is a 5 prime because this is the opposite orientation from this side, and this is the 3 prime. Okay, so this DNA polymerase is going to add a C here. It's going to add a G here. It's going to add a G here. It's going to add an A here. It's going to add a T here. Okay, and so there are nucleotides just floating around in the nucleus, and these nucleotides come from food that you eat. So everything you eat comes from a living organism, and every living organism has DNA. So your body will break down the DNA that you eat into nucleotides. They're just floating around, and then DNA polymerase will grab them and build new DNA from them. So when you need to make new DNA, you have nucleotides floating around to make new DNA. Okay, so I want to point out a couple of things. One is that this DNA polymerase built this new DNA strand, which is blue, 5 prime to 3 prime. This is very easy because this DNA here is being unzipped going up. Okay, so it's going to continue to open up. Helicase is going to open it up, unzip it, and this DNA polymerase is just going to follow the helicase, no problem. This is no problem. So this side here is called the leading strand. Okay, so the leading strand is the new DNA strand where the DNA polymerase follows the helicase. Okay, the other side over here is going to be being built by DNA polymerase also. And this one is going to be built 5 prime to 3 prime. So it's going to be being built in the opposite direction. Okay, so let me draw that. So on this side, the DNA polymerase has to start here. Okay, so I'm just going to draw it here. I know this might get messy in your notebook and you can't erase the way that I can. But I just want you to see what I'm talking about, okay? This is the DNA polymerase, and it's going to move from this direction, going backwards, okay? So helicase unzipped it. This is the replication fork. DNA polymerase is going to start here and move in the opposite direction of helicase. So the new strand, I'm going to make it blue. I'm going to put it here, is going to go in this direction, where this is the 3 prime end and this is the 5 prime end, because DNA polymerase can only work from 5 prime to 3 prime. So here is going to be a T, this is going to be a C, this is going to be a C, this is going to be a G. Okay, up here it was an A, but it's all blocked right now. But I just want to point out that we have CG over here on the left, CG on the right, CG on the left, CG on the right. I'm sorry, GC on the bottom. CG here, CG here, TA here, TA here, AT, AT. So you have 2 strands that are identical. Okay, but before I write that down, I just want to point out that this strand that we just drew that's going in the opposite direction is called the lagging strand. The lagging strand is the new DNA strand where the DNA polymerase goes in the opposite direction of the helicase. Where the DNA polymerase goes in the opposite direction. To helicase. And because it is going in the opposite direction, it is making the DNA in fragments. Okay, so what happens is this DNA polymerase on the lagging strand is going to start right here where the replication fork is and move down. Once it moves down here, it then has to wait for this part of the DNA to open up. So once helicase opens up this part here, let me just once the DNA helicase opens up this part, then this DNA polymerase is going to jump up here and move downward, okay? Make another fragment. And then the DNA polymerase is going to jump up here and move downward. So it has to make the DNA in fragments, okay? I know that's kind of messy. I'm going to erase it because it's so messy, but I just wanted you to see what I'm talking about, okay? And so those fragments have a special name that are called Okazaki fragments. So I'm going to write this down. Okay, DNA, this is for the lagging strand. DNA is made in fragments called Okazaki fragments. Okay, that's the name of them. And it's named after a Japanese scientist who figured this out. And it was kind of a big deal because everyone was always like seeing the fragments and thinking that there was a mess up in the lab technique. And they were just like, every time I do this lab experiment, there's always these DNA fragments. And then finally, this Japanese scientist brilliantly discovered that that's part of the DNA replication process, is one side is made in fragments because it can only build DNA from 5' to 3'. Okay, so it has to go opposite. Okay, so we have the leading strand, which is making one long continuous new DNA. We'll have the lagging strand where the new DNA is being made in fragments. Okay, and so the last step is where all those fragments need to be glued together. Okay, so that's the last step. And that's done by an enzyme. Whoops. That's done by an enzyme. This is step 4. And this enzyme is called ligase. So ligase is the 4th enzyme. This enzyme glues together all the Okazaki fragments. Oh my goodness. Okay. And so the last point that I want to make is that, as you can see here, we start off with one DNA molecule, and now we end up with 2, and they're identical. But I want to point out that half of the new molecule, so there's 2 identical molecules, half of this molecule is old and half is new. And in this molecule, half is old and half is new. And so this is actually called a semi-conservative replication because it's not that the new molecule is all the way new. It's only half new. It's half old and half new. So every piece of DNA has half of an old piece, which is called the parent strand, and then half of a new piece, which is called the daughter strand. But I just want to write this word down that this is called semi-conservative replication. Okay, and that means that it's not all the way new, it's only half new, okay? Okay, so that's it for today. I hope everything's going well, and I will see you next time. Bye-bye.